Thanks a lot for inviting me, and I'm
Thank
very
you.
proud that I can tell you something about the Insignia EU action. I hope you still hear me.
Yes, we can
Yes.
hear you and we see the presentation.
Oh,
Perfect.
okay,
Go
okay.
ahead.
Thanks a lot. Thanks a lot. Let me explain briefly what it is about. The Insignia EU study is an action of the European Union to use the honeybee colony as a biomonitoring of environmental pollution. So it's that simple, and we checked the colonies for pesticides, microplastics, air pollution, heavy metals, and pollen diversity. And to be clear, the study was conducted in all the 27 EU countries in cooperation, of course, with the Insignia beekeeper citizen scientist. Without the beekeeper, it would be impossible. And the Insignia Consortium, you see them listed below, and 630 honeybee colonies who actually did the work. And before I will present you the results, I want to show you and explain how the honeybee colony is acting as a biomonitor. And basically, it's the honeybee who collects all pollutants that are airborne on the plants, as you can see here on the left drawing. And they pick all this stuff up when they are collecting their food. They bring it to the hive, and the amount that every single honeybee collects is not measurable. It's so little that you cannot measure it. But they accumulate all this stuff in the honeybee colony to measurable accounts. But still, it's in the honeybee colony. And then we need the beekeeper citizen scientist here to collect these pollutants from the colony. And how does he do this? He does it in an in-hive passive sampler. That means that no bee will be killed or anything of the colony will be disturbed. To show you briefly, and then I go to the results, we use the AP strip, which collects the pesticides that enter the colony. We use the AP trap, which collect the microplastics. We use the simple silicon band to collect the air pollutants, propolis for the heavy metals, and honey for the polar pesticides. Here, the apiaries where we sampled it, so you can see it's all over the European Union. And we have the forest, the agriculture, and the artificial. Artificial means cities, urban regions. And we divided the apiaries over those three dominant land uses. So let me start by pollen diversity. It's a bit strange because pollen diversity is not an air pollutant or not any pollutant at all, but it is something for the beekeeper to see what the diversity of pollen is that there are for their bees. And we collected every 2 weeks the pollen in a pollen trap. All together, we had over 2,500 samples, and they were analyzed by DNA metabarcoding, the ITS2. What we did see is that all over the European Union, the pollen from the Astraceae, the Brassicaceae, the Fabaceae, Rosaceae, and Plantagenaceae are very, very much present. Here, I refer to the agriculture and the natural and artificial, so the land uses, and you see there's not that much difference in the number of genera that are collected there. And they are presented here. But you see the big gray bar, and this gray bar means that we found less than 3% of the pollen in this bar. That means that about, let's say 60% or even more, is collected in very low amounts and is the result of foraging for scouting flights. And especially in the artificial landscape, which is urban, there are a lot of flowers, but not in that big volume and that big amounts. So you have a lot of pollen here in the gray area, which are present, but in very low amounts, less than 3%. And here below, which is more, we have detectable amounts of the salix, so the willow, papaver, rubus, trifolium, clover, raphanus, castanea, centaurio, compositae, plantago, and brassica. This is the overall result of the pollen diversity. I come to that later. Then I go to the insignia, to the polar pesticides. You know, pesticides do not enter the honey because they are lipophile, which means that when you have pesticides in your nectar and it's stored in the combs, most of the pesticides, almost all pesticides, go from the nectar into the comb. So the comb is a kind of clearing machine for the nectar, for the pesticides. And there are some pesticides which are not attracted to the wax, and they are the polar pesticides, especially the glyphosate, so the herbicide, the glyphosate, the Roundup. And we checked them for 8 of these polar pesticides, and we did find that in over 6% of all the samples that we had, and you can see here over 1,100 samples, we analyzed them, and 4 pesticides were found, and they were all glyphosate or derivatives of glyphosate. So in 6% of the samples, we do have detectable amounts of glyphosate, all below the MRL, but it's there. And it's in line with the European food security, which also checks this in other food products and find more or less the same percentage. Then I move over to the metals. Heavy metals are everywhere. They are needed for the bees. They are biological important. But there are some metals, most like the synthetical ones, the new ones, and also metals in high concentrations, which are a problem. You know, in the biological frame, we need these metals as a part of our enzymes, etc., and protein. They have a biological function, but once there are too many and the concentrations are too high, they get toxic. Well, what we did find is this graph, this here, and here you see that the red line is the European average, and we have more or less here the same per land use. So the agriculture have a bit more over the European average, the red line, and the rest is a bit below it. It's quite normal and it's everywhere. But we do see a difference between the north of Europe and the south of Europe. You see here the dark diamonds, and here the green bars, which means the green bars are the values in the north of Europe, and the black diamonds are the European average. And in the north, it's all below the European average, and in Greece, in the south of Europe, it's all above the European average, which is quite normal. And this is because the natural soils in the south of Europe have, by nature, more minerals and more metals. So this is a reflection of what is normal in nature. Then we come to the silicon wristband. The simple silicon wristband is highly effective in absorbing this air pollutants, and then we're talking about the polycyclic aromatic hydrocarbons and the volatile organic compounds. This sounds rather difficult, but I can tell you that, for instance, these volatile organic compounds are very, very natural. For instance, what we have in our colonies, the green substance and all the pheromones that the honeybee inside the colony is communicating are volatile organic compounds. There are many, many of these compounds, but we selected the VOCs and the pHs for the compounds that are hazardous to life, so the synthetic ones that really poses a hazard to us and to the bees. We did find them everywhere, also the hazardous ones. And the highest concentrations that we found were benzene, isoprene, hexane, and naphthalene. And they are both natural and both synthetic. Here we have the number of land use, and then you see the land use doesn't matter at all. It's air pollutant, air pollution, and air is everywhere. It's always refreshed and disturbed, and there's always wind. So the levels in the air above the agricultural landscape, the urban and the forest, it's all the same, which we also expected. All the target compounds of the VOCs and the pHs were detected. Brings me to the microplastics, which is a big problem nowadays in the modern world. We have a lot of plastics, and we're still producing a lot of plastics. And we developed a method, this in-hive sampler, this AP-trap, which is a sticky board here, the white one, which is in the cage, so the bees don't touch it, but the air is flowing around in the hive. There's a constant airflow inside the hive, stimulated and continued by the honeybees to refresh the air and to keep the temperature down. And on this way, there's also always a airstream inside the colony, which contains microplastics. And also the bees have microplastics on their body, which they collected from the flowers. So this AP trap, this sticky board, collects and traps the microplastic particles. And to know, because we're talking about microplastics and what is microplastic, here is an example. This is how microplastics look like. They are fibers, they are fragments, they are films. That's all about the morphology of the particles. And they are in all kinds of colors. And this is what you have in your colony. And we detected a lot of them, over 50,000 fibers. And here we have, and I show you here the fibers per land use, and it's more or less the same in all land uses because microplastics are everywhere. In the city, a bit more, in forest, a bit less, but more or less is it the same everywhere. To know what kind of microplastics that we are dealing with, we have the micro FTIR, this is the Fourier transformed infrared spectrometry. It's a complicated method, but it's possible to take such a fragment and such a fiber, put it in the microscope, on the microscope, and use this method, this infrared spectrometry, and then you see this pattern that shows you this is a pen, this is cellulose. Here we have A, which is all abbreviations of a certain microplastic. From PET is a very known one, and the other ones as well. So this is the result, and we see here it's omnipresent there all over Europe. In all apiaries we do find microplastics, and we don't see that much difference between the apiaries. There are some differences between countries, which mostly had to do with the location. For instance, in Ireland, there's always a lot of wind. You don't have that many than, for instance, in Poland. Then I bring you to the pesticides and biocides, which is, of course, the most interesting, at least for the beekeeper. And to do this, we developed the AP strip, which is really revolutionary, and it's now applied all over the world. It's a simple strip, and we dissolved and attached Tenax. Tenax is a plastic, it's a polymer which is used in the laboratory for spectrometry, but it also can be applied on the strip. So it's a procedure which is public, and everybody can produce this kind of AP strip, which is really effective in binding all kinds of pollutants. not only pesticides, but we focused in this on pesticides. We had 315 apiaries here. These are 315 dots. Don't count them, but they are 315. Altogether, we analyzed about 6,000 api strips, which is a lot of work, and we detected over 200 pesticides, which is a lot. Here on this graph, you see that agriculture, artificial and forest, so this is urban. We see that here in agriculture, you have about 4 pesticides per sample. So every sample has a medium of 4 pesticides. And there is no apiary in the European Union which does not have a pesticide. So every apiary is facing and is exposed to pesticides. So here we have the medium, which is 4 for agriculture, and the top is about 25. Looking at artificial, which is the urban region, we have about 3, but we have up to 29 pesticides in some samples per sample. So this is a lot. And also in the forest, you have, even where you think this is a national conservation area, you do have pesticides because pesticides are taken away by the wind. There's always a kind of dust from the soil. A lot of pesticides, most of them end up on the soil because when you spray, most of them is ending up on the soil. Then when there's warm weather, they have dust and it's all spread far, far away. So this is the result. There is no place in the European Union, and I think over the world, where you don't have pesticides. Here is the number of the detections that we have, and this is the percentage of the total land use. And you see most apiaries were in the agricultural land use, about 70%, and that is because bees are by nature, and beekeepers are by nature in agriculture areas. So this is a normal distribution, and we could show these pesticides. With all these data, we were able to make predictive models for plant protection, pesticides, and you see here what is changing from May till September, August, September, and then again starts in May, and the same for pesticides here. So this is what we have. And then I come to the conclusions, and I can talk about it for hours, but this is a restricted number. So pesticides, we can say they're omnipresent. Everywhere are pesticides, and bees may be exposed to a mixture of 28 pesticides at the same time. We also have a lot of non-legislated compounds, and and old compounds like DDT, which is already forbidden for decades, but still find a lot of DDT. We find a lot of imidacloprid. We find a lot of fipronil. So we also have a lot of these pesticides. The pesticides in the nectar, as I told you, over 6% of all the honey samples has glyphosate or a derivative of glyphosate. Microplastics are everywhere. In every honey, in every colony, you find microplastics. Same for air pollutants. In every hive, but that means also in the ambient air around the hive, you have this air pollutants, which is everywhere. But we could detect some places where it's more than on other places. So where these air pollutants are produced due to natural conditions like forest fires, but also in factories. Heavy metals, of course, we have a natural background. They're normal in nature, but we could see that there were emission sources where there is much more than we expected. So it's produced there, and then we're talking about factories, heavy factories, steel industry, etc. And pollen, we found a lot of pollen. Most of what we did find is less than 3% as a result of the scout flies. And there's some good foraging crops from Astraceae, Rosaceae, Brassicaceae, Plantagenaceae, and Fabaceae, which was already known. It's in beekeeping. We made these predictive models. So I can conclude that the INSIGNIA developed a non-invasive biomonitoring citizen science methodology. It's a long word, I know, but it's important to mention non-invasive biomonitoring citizen science methodology to biomonitoring the environmental pollutants with honeybee colonies. Now, we have completed the study. We did one in 2020, we did one in 2023. So we hope that the European Union will allow us to continue this biomonitoring, because the more that you repeat it, you see some trends. And we advised the European community and European Commission to expand it to a more ecotoxicological approach, so that we can see what's the impact of this pollutant on life. Because what I just presented you is qualitative, so we could see it's yes there and none there, but it's also possible to have in more detail and see is there any impact on life, and in this case especially on the honeybees. There's something else which is also very important, because we faced a lot of pathogens from the veterinarian world, which is also that are pathogens for humans as well. And most of them are airborne, and I can, or for the veterinarians, like swine fever and more an avian flu, and they are spread by airborne, and you can see them in the eDNA on the very first, very low amounts. So when we use the pollen that we are collected to see the biodiversity, you also can check them for this kind of pathogens and use it as an early warning. Just to show off, we have a lot of publications. We have 60 up till now, and there are more coming. The end report will be published soon on the EU publication site, and it's on the INSIGNIA website. We have a guideline, a new guideline, which was written last year, and it has all the information you need as an institute or whatever to perform this kind of studies. It has all the data on how we do it, on the picture manual, so the instructions for the beekeeper. It has all the laboratory protocols, so everything you need for a proper biomonitoring system is written there and is publicly available. And we also have the brochure, in which is in simple language for the not to use, but it gives a lot of information. And once again, I tell you, this is really the first study in the European Union where on the same day or the same range of days, same weekend, the same sampling was done all over Europe, which was quite an organization, but we managed. So from Finland, the north of Finland, till the south of Greece and Cyprus, we all did the same. We collected the api-strips, we collected the api-trap, we collected the silicon bands, we took the propolis, we took the samples from the pollen, all in the same day, which gives a huge amount of information, which is now publicly available.